So last week we finished reading Chapter 7 in the textbook and watching the videos for Chapter 7. This week I'd like to recap what we learned in Chapter 7 because it is pretty well known to be one of the most difficult concepts in biology and biochemistry. So remember, Chapter 7 was about cellular respiration, how we harvest the potential energy stored in the chemical bonds within glucose using oxygen to produce energy, or ATP. And once we oxidize glucose to release that energy, we produce 6 carbon dioxide molecules and 6 water molecules. The main goal of this week's homework assignment is to fill out this table, and you will post it on a discussion in order to summarize the learning from Chapter 7. We really care about the reaction or purpose of each of these steps, what goes in, and not every single molecule or compound, but we're really looking at the main players that go in, what comes out, where these reactions happen in prokaryotes and eukaryotes. Remember, prokaryotes do not have a nucleus or organelles that are membrane bound, like the mitochondria would be lacking in prokaryotes. Eukaryotes, like us, humans, do have a nucleus and do have organelles like mitochondria. And then we're also going to look at special requirements or interesting byproducts of these. So what we're going to look at is start with glycolysis. And I remember the purpose of this was to take a 6-carbon glucose and break it into 2 3-carbon pyruvic acid molecules. And remember, that did not require oxygen. If oxygen is available, the next 3 pathways will happen. But if oxygen is not available, we're going to see that right after glycolysis, you're going to go into fermentation, which is not oxygen dependent and allows you to regenerate the NAD+ necessary for glycolysis to continue. Let's see, here I have a bunch of different products that can be generated from fermentation, but in our book we really mainly focused on lactic acid production, such as in humans, also known as lactate, and ethanol that's generated during fermentation in yeast. I do want you to know that other products are possible depending on what kind of organism you have. So certain bacteria, when they ferment, they produce these byproducts. All right, so the homework will be to fill out this chart by watching the rest of this video. I go through the entire chart with you. And also look at the pictures that I've hand drawn that are attached to the document, the homework, to see if you can kind of put things together. So let's look at those 2 pictures. All right, so let's summarize Chapter 7 as it occurs for eukaryotes. Here is my equation for cellular respiration, and here is a giant cell that I drew that's not to scale. I did not draw any organelles that are not relevant directly to cellular respiration. So this is the cytoplasm, so this must be the cell membrane, plasma membrane. And I only drew 1 mitochondrion, and I made it huge, so it's definitely not to scale, in order to kind of fit all of the reactions into the mitochondria. So glycolysis happens first, and that makes sense that it happens in the cytoplasm because that's where I get my glucose transported into the cell. Glycolysis chops it in half and produces some of the ATP and NADH we saw earlier, and no oxygen is required directly. So if there's no oxygen available, the next step is fermentation to produce different types of products depending on what kind of organism you're looking at. If there is oxygen, then we see all of these reactions happening in the mitochondria, including the production of acetyl-CoA, the Krebs cycle, also known as the citric acid cycle, and then the electron transport chain and the production of ATP through chemiosmosis. But if we are looking at prokaryotes, this will be different. So let's look at prokaryotes in the next slide. All right, here is the same thing happening in prokaryotes that do not have a nucleus or mitochondria because they don't have any membrane-bound organelles. To orient us, this is the cytoplasm of the cell, and I see that there's no mitochondrion. So I start with glycolysis, as I did earlier, fermentation if there's no oxygen. Right after that, if there is oxygen available, I can go through the Krebs cycle, and the electron transport chain, instead of being embedded in the inner mitochondrial membrane, these proteins are embedded in the plasma membrane. So I can see that protons are shuttled out of the cell to build that gradient needed to produce ATP through chemiosmosis. All right, so in the next couple of slides, I'm going to go through that table step by step so you can fill it out to submit your homework, and then you'll be done for this assignment. Okay, there's a blank one here in case you want to practice, and you should have one in the slideshow version, the PDF version of your slides too. This is a chart we would generally hand out in our face-to-face course. It's a nice way to summarize this chapter because there are so many reactions happening. But I would go through this chart and see if I can figure out what's the purpose of each of these reactions. Glycolysis, what's that purpose? I'm going to chop up glucose, remember 6-carbon glucose, into 2 3-carbon pyruvic acid molecules. So I've got pyruvic acid, and I remember that had 3 carbons each, and I made 2 of them. I had a net of 2 ATP, a net of 2 NADH. It happens in the cytoplasm of both. So I don't need oxygen here, and remember the method of making ATP for glycolysis was substrate level phosphorylation, where we're literally stealing a phosphate group from another molecule and sticking it onto ADP to make ATP. If we're going to go through respiration, these are our next 3 steps: synthesis of acetyl-CoA, Krebs cycle, and electron transport chain. So in those cases, then that would happen right after glycolysis. I can see what are the purpose of these. Synthesis of acetyl-CoA is to turn a 3-carbon compound into acetyl-CoA, a 2-carbon compound. Krebs cycle, I'm making a lot of high energy electron carriers like NADH and FADH2. I'm also getting rid of the carbons. So in the Krebs cycle, you're going to see you get rid of the carbon dioxides. This is a continuation of that previous slide, but I wanted to erase this part, and I erased everything on accident. TCA stands for tricarboxylic acid cycle, which is an old name for the Krebs cycle. I wrote it just in case you've heard of that before, but don't worry about it if you haven't. Another main goal of the Krebs cycle is to oxidize the remaining carbons found in the acetyl-CoA. So oxidize remaining carbons. And then I had these blank earlier. Synthesis of acetyl-CoA. This is in the cytoplasm for prokaryotes. And the eukaryotes, this is on the mitochondrial membrane, which is the cristae. So that inner membrane of mitochondria. For the output of the Krebs cycle, we've got 2 ATP, 2 FADH2, oops, that one is 6 NADH, and this is 4 CO2, and this is per glucose, not per turn. Where does this happen? This is the not this side. On the right side, this is the mitochondrial matrix. So that liquid portion of the mitochondria, and in prokaryotes, it's still the cytoplasm. For the electron transport chain, their purpose is to regenerate the NAD+, and we have it really here, and the FAD. We also make a lot of ATP, lots of ATP, through oxidative phosphorylation, also known as chemiosmosis. So where does this happen? In prokaryotes, it's on the cell membrane, or you could say plasma membrane. In eukaryotes, this is on the mitochondrial cristae again. Remember that inner membrane, the folded membrane. These are where those enzymes are located. Fermentation is the last one. So again, these 3 were all part of cellular respiration. Glycolysis is the first reaction, regardless if it's respiration or fermentation. And if we don't have oxygen, we can also go through fermentation, which is going to help us regenerate NAD+. And you could put that purpose over here. Where does this happen? Cytoplasm of both, just like glycolysis. All right, so at this point you should have everything filled out on your chart, and you can print out extra copies of this, blank ones, in the document I gave you in case you'd like more practice, or even take a blank one out and see if you can say it out loud what's in the missing areas. And let me show you how you're going to submit this. All right, so when you're done with the chart, you're going to go to the Week 7 module on Canvas and find the Chapter 7 discussion. You'll see some directions at the top of the page, but one of the things you're going to do is take a picture of the chart that you just finished and embed it into your post. So I just wanted to show you how to do that. You're going to click on this icon that looks like a little sun and mountain. That will allow you to embed your picture within your post. So it shows up here, and we don't have to click on a separate link or download it. We'll just be able to view it on the discussion page. All right, feel free to email me if you have any questions in the meantime, and I will see you guys again soon.